Have you ever stopped to consider that something really common, like a lingering viral infection, maybe one you didn't even notice, could be subtly, secretly connected to something as serious as life-altering as cancer?
It does sound a bit like science fiction, doesn't it?
It really does. But what if I told you that actually a significant percentage of human cancers aren't just random cellular mishaps, not just independent events, but are actually tied, often quite deeply, to specific viral infections?
Yeah, it's a surprising connection for many people.
So today on The Deep Dive, we're taking a truly fascinating journey into the world of oncogenic viruses. These are tiny, often very stealthy biological entities that possess this remarkable ability to cause cancer. Our mission today is really to unpack precisely how these microscopic agents can fundamentally alter our cells, turning them from healthy parts of our bodies into something malignant and ultimately leading to disease.
And we've got a great set of sources to draw on here to really illuminate this incredibly complex and frankly vital interplay between viruses and cancer.
Absolutely. And the scale of it is just, well, it's staggering when you first hear it.
It really is. What's truly illuminating here is that sheer scale. We're not talking about some rare occurrence or a, fringe theory. The data from our sources, it reveals that approximately 10 to 20% of all human cancers are, in fact, directly attributable to oncogenic viruses.
Wow.
Think about that for a moment. That's a substantial portion of cancer diagnoses, a staggering number that many people might not even realize has a viral origin. It truly highlights this unseen sort of silent battle happening right within our bodies, one that science is increasingly bringing into focus.
10 to 20%. That is profound, much higher than I think most people would guess. So, okay, to make sure we're all on the same page, let's just start with the basics.
Yeah.
What exactly do we mean by oncogenic viruses?
Yeah, good starting point. At its core, an oncogenic virus is simply a virus that has the capacity, the potential to produce tumors in a host. The fundamental concept to grasp here is transformation.
Transformation.
Okay. Right. This refers to the profound process where these viruses convert a normal, healthy cell into a malignant one. It's not just a subtle shift. It's a fundamental change in the cell's very nature, its internal programming, its behavior, often leading to uncontrolled and abnormal growth.
Like reprogramming it.
Exactly. It's like rewriting the operating system of a cell to make it go rogue.
And crucially, this isn't something that happens overnight, is it? Our sources kept mentioning this idea of a long latency. What does that mean for how these viruses actually work inside us?
That's a really critical point. And the concept of long latency is particularly intriguing, almost like a ticking time bomb in a way. It means there can be a significant time gap, not days or weeks, but often years, sometimes even decades.
Decades, wow.
Yes, decades between the initial viral infection and the eventual development of a discernible tumor. This extensive delay makes detection incredibly challenging, and it raises profound questions about prevention strategies, too. Imagine getting infected with an oncogenic virus in your youth and only experiencing cancerous changes much later in life. It's a silent, persistent threat that our immune system often struggles to fully clear.
So we're embarking on this journey to understand these, patient, persistent viruses that cause cancer, often after this long, silent period. What exactly will we explore in this deep dive to really get a handle on their impact?
Well, we're going to journey deep into the microscopic realm, right into the cellular level itself, to unravel precisely how these viruses work their intricate magic. Or maybe their profound mischief is the better word.
Right.
We'll examine the delicate balance within our own cells, those regulatory systems that keep us healthy, and how viruses can hijack them. Then we'll look at specific prominent examples of these viruses. We'll, meet them by name, uncover the particular cancers they're associated with. Following that, we'll shift our focus to the scientific pursuit, discussing how these viral cancers are diagnosed in the lab, what current treatment approaches look like, including the challenges and the breakthroughs. And finally, in a truly surprising twist, we'll uncover their significant implications even for something as, routine, yet vital, as your dental health.
Dental health, okay, that's intriguing.
Yeah, it's a comprehensive, really eye-opening look at an often overlooked dimension of cancer.
Okay, let's unpack this then. Dive right into the truly microscopic yet profoundly impactful battleground, our own cells. Before a virus can even start to make a cell go rogue, we need to understand how our bodies normally keep everything in check, right? Healthy.
Exactly.
Our bodies have this incredibly sophisticated system for managing cell growth and division. It's meticulously controlled by specific genes. You can really think of it like a finely tuned orchestra, where each section, each type of gene has a crucial, precise role in maintaining cellular harmony, preventing chaos.
That's a great analogy. The cellular growth and proliferation is a tightly regulated dance, and it's managed by 4 primary categories of genes. When any of these regulatory genes experience a defect, or are somehow subverted perhaps by a virus, it can lead directly to that transformation of a normal, healthy cell into a tumor cell.
A delicate balance.
Very delicate. An intricate set of checks and balances that, if disrupted, can have, well, catastrophic consequences.
So let's start with the first category, the ones you might think of as the accelerators for cell growth. Proto-oncogenes. What's their normal job, their healthy function, and how do they go wrong when, say, a virus gets involved?
Right. Proto-oncogenes are absolutely fundamental for life itself. In their normal, unmutated state, they act as the essential accelerators for cell growth and proliferation, promoting healthy, controlled cell division.
Controlled being the key word.
Absolutely. They're vital for our development from a single cell, for repairing tissues after injury, for the everyday replenishment of cells throughout our bodies. Indispensable. However, when things go wrong, often through mutation, or, as we'll see, viral influence and overactivation of these genes can occur.
The accelerator gets stuck.
Pretty much. It essentially means the accelerator gets stuck in the on position, leading to uncontrolled proliferation and the transformation of normal cells into tumor cells. This is a critical insight. Something that is vital for life, when unregulated, can become profoundly harmful. It just highlights how exquisitely balanced our cellular systems really are.
Okay, the accelerator analogy makes perfect sense. So if proto-oncogenes are the accelerators, then the next category, anti-oncogenes, often called tumor suppressor genes, they must be the brakes.
Precisely. Anti-oncogenes are indeed the brakes, or if you prefer, the quality control checkpoints of the cell. Their normal function is paramount. They continuously monitor cellular growth, acting like vigilant guardians to suppress any abnormal or runaway proliferation.
Any famous examples?
Oh yeah. Classic and well-studied examples include the retinoblastoma, or RB gene, and perhaps even more famously, the P53 gene, often called the guardian of the genome.
Right, I've heard of P53.
It's crucial. If these vital genes are inactivated or lost, or their function is compromised, which, by the way, viruses are quite adept at doing the cellular brakes fail completely, this then permits abnormal cell events to occur unchecked, significantly contributing to cancer development. It's like trying to stop a runaway car when the brake lines have been cut.
Okay, so accelerators and brakes. But what about, the general wear and tear on our DNA? There must be a repair crew for our genes, too.
Indeed we do. A vital function performed by the DNA repair genes. Their normal function is absolutely crucial. They are the dedicated repair crew for our genetic material, diligently fixing the countless mutations and damages that inevitably occur during cell division, or from environmental factors like UV light.
Our DNA is constantly getting hit.
Constantly undergoing minor damage, yes. And these genes are our internal error-correcting mechanism, ensuring genomic integrity. However, when things go wrong, when these repair genes fail, damaged DNA goes unrepaired. This leads to persistent mutations that can directly drive cancerous changes. These accumulated errors, like compounding interest, significantly increase the risk of malignant transformation over time.
Makes sense. And finally, we have genes that control cell death itself. That sounds a bit dark, but I guess it's essential.
Absolutely essential, not dark at all. It's vital. These are the apoptosis regulatory genes.
Apoptosis?
Apoptosis is the term for programmed cell death, and it's a graceful, meticulously orchestrated, and absolutely necessary process. It's where old, damaged, or simply unneeded cells are efficiently eliminated from the body. These genes precisely manage this process. They either upregulate it to initiate cell death or downregulate it to allow cells to survive as needed. It ensures cellular turnover and overall tissue health.
So why does it matter so much in the context of cancer?
The critical reason it matters is this. If these genes are dysregulated, meaning their function is impaired or they're overridden by, say, a virus, abnormal cells might not die when they should. Instead, they linger, they accumulate, they contribute directly to uncontrolled growth, eventually becoming part of a growing tumor mass rather than being neatly removed.
Like a faulty self-destruct switch.
Exactly. It fails to engage when it needs to.
Okay. So these 4 categories, proto-oncogenes, tumor suppressors, DNA repair, and apoptosis regulators, they all work together in this complex symphony to keep our cells orderly. But what happens when a proto-oncogene, that normal accelerator, gets mutated or, like we're discussing, influenced by a virus.
Yeah.
What does it become? What's the shift?
That's where we get the term oncogene. An oncogene is simply a mutated or abnormally expressed version of a proto-oncogene.
So proto-oncogene gone bad.
Essentially, yes. It's the accelerator stuck permanently on, constantly promoting uncontrolled cell growth and division. They contribute to cancer by fundamentally disrupting a cell's ability to control its own growth, making it ignore all the normal stop signals and growth constraints. It's a fundamental break in the rules.
And this is exactly where viruses come into the picture in this, well, really fascinating, almost insidious way. We mentioned viral oncogenes, or VONT. What are they, and how do viruses get these potent tools?
Okay, here's where it gets really interesting. The part that still kind of amazes researchers, some viruses actually carry their own viral oncogenes, VONT. These aren't just random viral genes. They are believed to have originated from our own proto-oncogene.
They stole them from us.
In a way, yes. Over vast evolutionary time, through complex processes like recombination, viruses essentially stole bits of our own cellular machinery, altered them, and weaponized them. It's like the virus scavenged a part of our cell's control panel and repurposed it for its own nefarious goals. These viral oncogenes then encode specific proteins that directly trigger the transformation of normal cells into cancer cells. They are, in essence, ready-made cancer genes that the virus brings with it, or they encode viral oncoproteins that precisely influence cellular growth control. They promote growth, prevent apoptosis, override limits on DNA synthesis. It's a direct, sophisticated assault on the cell's entire regulatory system.
Incredible evolutionary adaptation.
It truly speaks to the adaptive power of viruses.
Okay, that really sets the stage. We understand the battleground and the viral weapons. Now let's explore the actual viral strategy. How do these tiny things manage to turn a healthy cell malignant? It's not a quick hit and run, is it? It sounds more like a drawn-out subversion.
No, definitely not quick. It's a meticulously choreographed long game. For an oncogenic virus to successfully cause cancer, a specific multi-step sequence of events typically has to occur. It's a complex subversion strategy and intricate dance with the host cell over time. Our sources outline about 5 essential steps for this oncogenesis to unfold effectively. Think of it like a step-by-step invasion plan.
Okay, step 1. What's the foundational move in this viral long game?
The very first and absolutely foundational step is establishing a persistent infection.
Persistence.
Makes sense. Right. This isn't a hit-and-run viral visit where it infects, replicates, and leaves quickly. For oncogenesis, there needs to be a prolonged, intimate interaction between the tumor virus and the host cell. The virus has to stick around, integrate itself, continuously influence the cellular environment.
Like a squatter, not a guest.
Exactly, like a persistent squatter setting up permanent residence rather than a transient guest just passing through. This persistence allows those long-term cellular changes to accumulate.
Okay, so it decides to stick around.
Yeah.
But then it needs to deal with our body's amazing natural defenses. Step 2 must be evading the host immune response. This sounds like the biggest hurdle. How do they manage that?
It is a huge hurdle. Our immune system is incredibly sophisticated, powerful, designed specifically to identify and clear viral infections. So for an oncogenic virus to succeed, it absolutely must bypass these defenses, often with remarkable cunning.
What kind of tricks do they use?
Oh, they have a truly diverse and clever array of evasion mechanisms. For instance, some, like Epstein-Barr virus, EBV, especially in B cells, they restrict the expression of their viral genes so profoundly they become almost invisible, go largely unnoticed by immune cells.
Hiding in plain sight.
Kinda, yes, like whispering their presence instead of shouting, blending into the background noise. Other viruses, like human papillomavirus, HPV, use a different tactic. They infect sites that are relatively inaccessible to robust immune responses.
Like where?
The epidermis, that outer layer of skin, or mucosal surfaces. It's like finding a hidden hideout where the immune system's patrols struggle to reach them effectively. Allows them to establish long-term residency.
Clever.
Then you have viruses like HIV, notorious for rapid mutation. They undergo constant genetic camouflage of certain viral genes, changing their look just enough to escape detection.
An arms race.
It really is. An evolutionary arms race at the microscopic level, the virus trying to stay one step ahead. And finally, some viruses, again like HIV, directly infect and suppress essential immune cells themselves, like CD4 T cells.
The commanders of the immune response.
Exactly, which are crucial for coordinating the whole immune response. This effectively sabotages the very system designed to fight them, leaving the host vulnerable. It's truly a multi-pronged assault on our defenses, shows their remarkable adaptability.
That's quite a playbook for evasion.
Yeah.
But sometimes it's not just the virus being clever, right? It's also about a general weakening of our own immune system.
Yeah.
Step 3, immunosuppression.
Exactly right. Sometimes the host's immune system is weakened, maybe directly by the virus itself. HIV is the classic example of devastating immune cells. Or due to other factors, chronic illnesses, medication suppressing the immune system for transplants, even aging. This broader context of immunosuppression creates an environment where infected cells can proliferate unchecked. They escape the vigilant host immune response that would normally detect and clear them before they become cancerous. It essentially removes a critical barrier to tumor formation, allowing abnormal cells to gain a foothold and multiply. That's a key reason certain cancers are more common in immunocompromised folks.
And then there's the intriguing question of which cells are vulnerable. Step 4, host cell susceptibility. Are all cells equally susceptible? Is it a lock and key thing?
Definitely a lock and key situation. Not all cells are equally vulnerable, no. Some host cells are what we call permissive. They fully support viral growth and replication, producing new viruses. They act like factories.
Okay.
But here's the really fascinating paradox, the counterintuitive bit. Others are non-permissive. These cells might lack the right surface receptors for the virus to attach, or they just don't fully support the complete viral replication cycle. Ironically, the risk of oncogenicity, of cancer developing, is actually higher when a non-permissive cell gets infected by a tumor virus.
Wait, that seems backwards. Why would a cell that doesn't fully support the virus be more dangerous for cancer?
Yeah, it sounds completely counterintuitive, doesn't it? It's because in a non-permissive cell, the virus can get stuck in a transformative state. Instead of completing its full prolific replication cycle where it would normally boost the cell to release new viruses, destroying it in the process,
Right.
It integrates its genetic material into the host cell's DNA, and then it continuously drives the cell towards malignancy without actually killing it off. So instead of being a destructive transient invader, it becomes a permanent resident, a quiet saboteur.
Ah, I see.
This allows the altered cell to survive, multiply, and eventually become cancerous. It's a truly fascinating strategy for the virus. If it can't complete its life cycle, it settles for this more insidious long-term subversion.
So in these non-permissive cells, the virus becomes a permanent genetic fixture, which leads perfectly to step 5, retention of viral nucleic acid inside the host cell. How does that happen, and why is it so crucial?
This step is absolutely key for many oncogenic viruses. For them to persistently drive cancerous changes, their genetic material DNA copies, or RNA converted to DNA, must be integrated directly into the host cell chromosome. This makes the viral instructions a permanent, indelible part of the host cell's genetic blueprint. It gets passed down to every single daughter cell during division, ensures the malignant program continues.
So it's truly embedded.
Deeply embedded. Now, there is an important exception with retroviruses like HIV or HTLV. Their RNA genome actually gets reverse transcribed into DNA, first a unique process where RNA becomes DNA, and then this new DNA, the provirus, permanently inserts itself, a unique mechanism.
Okay.
And then there's a real anomaly with hepatitis C virus, HCV. RNA is neither reverse transcribed nor integrated into the host chromosome.
So how does it cause cancer then?
It's maintained within the tumor cells, but it causes cancer through long-term indirect mechanisms, primarily chronic inflammation and liver damage that drives continuous cellular changes and mutations over years, creates an environment ripe for malignancy.
And many different strategies.
It really highlights the diverse and adaptable strategies viruses employ to reach the same carcinogenic outcome.
Truly remarkable. Okay, now that we understand the mechanisms, the steps involved, let's move to the viral gallery. Specific examples of these oncogenic viruses and their cancer connections. How do we categorize these culprits?
Broadly, we categorize them into two main groups based on their genetic material: DNA viruses and RNA viruses. This fundamental distinction helps us organize and understand them, as their genetics often dictates their strategy and how they interact with our cells.
Makes sense. Let's start with the DNA viruses. They seem to have a strong presence. Who are the major players?
First up, and probably one of the most well-known, are the human papillomaviruses, HPVs. These are small, non-enveloped DNA viruses, icosahedral shape. They're well known for causing benign things like common warts, oral papillomas.
Right, warts.
But their darker side is their strong link to the progression of various cancers, specifically high-risk types like HPV 16, Negist 18, Negist 33. They're frequently and strongly associated with invasive cervical cancer, a huge public health concern globally. Furthermore, types 16, 18, 33, and 35 have also been definitively tied to oral premalignant lesions and oral squamous cell carcinoma.
That can be cancer too.
Yes, and their mechanism is quite direct, quite aggressive. Their viral E6 and E7 proteins act as powerful oncogenes. They directly interfere with and essentially disable our host regulatory proteins like P53 and P16, basically cutting the cellular brakes, letting the accelerator run wild.
Directly sabotaging critical defenses. Wow. What about herpes virus? We hear about those a lot. Cold sores, chicken pox. Are they oncogenic too?
Herpes viruses are indeed another major family of DNA viruses, and several members have oncogenic potential. These are large, enveloped DNA viruses, also icosahedral. The most prominent oncogenic one is Epstein-Barr virus, EBV, a remarkably widespread virus. Most of us get infected at some point. It's strongly associated with several cancers, Burkitt's lymphoma, particularly in parts of Africa, and nasopharyngeal carcinoma, more prevalent in Southeast Asia. EBV has this remarkable ability to transform B cells, immune cells, into immortal cells that can lead to malignancies. It's also linked to Hodgkin's and non-Hodgkin's lymphomas, shows its broad oncogenic reach.
And there are others in that family.
Yes. Then there's human herpes virus 8, HHV8, also known as Kaposi's sarcoma-associated herpes virus, KSHV. This one is directly associated with Kaposi's sarcoma, a cancer in the lining of blood and lymph vessels, particularly common and aggressive in immunocompromised individuals like those with HIV/AIDS.
Ah, the link to HIV again.
Yes, it's often a common oral manifestation of HIV infection, sometimes the first sign. KSHV is also involved in other lymphomas in AIDS patients, again underscoring that link between immune suppression and viral cancer. Finally, herpes simplex virus type 2, HSV2, known for genital herpes, is considered a potential co-agent in some variants of cervical cancer, often working alongside HPV.
Quite a rap sheet for the herpes virus family. What about hepatitis B? That's a major global concern.
Absolutely. From the hepadnavirus family, we have hepatitis B virus, HBV, small spherical DNA viruses. They have a unique characteristic. They synthesize their DNA by copying an RNA intermediate using reverse transcriptase, usually associated with RNA viruses.
Interesting.
HBV is a recognized and significant cause of human hepatocellular carcinoma, primary liver cancer. Chronic HBV infection leads to persistent inflammation, liver damage, creating an environment where liver cells are prone to cancerous changes.
Okay. And adenoviruses. I know they cause common things like colds, pink eye. Are they oncogenic in humans?
That's an important distinction and a great question because their lab behavior can be misleading. Adenoviruses are medium-sized, non-enveloped DNA viruses. They affect eyes, respiratory tract, GI tract, urinary tract. About 47 types, often asymptomatic, common in kids. They can have 3 interactions: infection, cell damage, latent infection, hiding, reactivating, and yes, oncogenic transformation in vitro in the lab.
But not in people.
Right. And this is crucial. While they can induce tumors in newborn hamsters in lab settings, no association with human neoplasms has been found. This is a vital caveat, a testament to the complexity of viral pathogenesis in the real world. Not everything that transforms cells in a dish causes human cancer, shows the difference between a lab and a complex human body.
That's a really important reminder. Lab results aren't always the whole story. Okay, let's switch to the RNA viruses. What do we need to know there?
Among RNA viruses, the retroviruses are significant players. This group includes human T-cell leukemia viruses, HTLVs, and also, of course, HIV. Large, spherical, enveloped RNA viruses. Unique because they have that enzyme, reverse transcriptase.
Right, the RNA to DNA enzyme.
Exactly. It lets them transcribe their RNA into DNA. This DNA then integrates into our host genome, forming that provirus, a permanent viral blueprint in our chromosomes. HOV is a prime example. Spherical, enveloped 120 millimeter, 3-layered structure, a master of stealth and integration. Specifically, HTLV-I is the primary agent of adult T-cell leukemia lymphoma, ATLL, a very aggressive cancer of the immune system.
And how does it cause the cancer? Indirectly.
Yes, these viruses often promote cancerous growth indirectly using a specific viral gene called the TAX gene. Instead of carrying a direct oncogene, TAX acts more subtly. It enhances the expression of cellular growth genes and interferes with tumor suppressor pathways, driving uncontrolled cell proliferation. A clever, indirect manipulation.
And hepatitis C, another RNA virus often linked to liver issues.
Yes, from the Flaviridae family, hepatitis C virus HEV. An RNA virus, single-stranded RNA genome. It's a significant cause of primary liver cancer, hepatocellular carcinoma globally, and as we mentioned earlier, HCV is unique.
Because it doesn't integrate.
Right. Its RNA is neither reverse transcribed nor integrated. It causes cancer through those long-term indirect mechanisms. Chronic HCV infection leads to persistent inflammation, oxidative stress, repeated cycles of cell death and regeneration in the liver. This prolonged turmoil results in accumulated genetic damage, cellular instability, and ultimately cancerous changes over many years. A classic example of chronic inflammation driving malignancy.
Incredible. Not just the diverse mechanisms, but even the physical appearances of these tiny agents vary so much too. It helps to picture them.
Absolutely. Their morphology, their physical look is as diverse as their genetics and mechanisms. Papillomaviruses like HPV, small, non-enveloped icosahedral, that 20-sided geometric shape. Herpes viruses, much larger, enveloped, also icosahedral. Hepatinoviruses like HPV, small and spherical. Retroviruses, HIV, HTLV, large, spherical, enveloped, often with that distinct three-layered structure under a microscope.
And the giant.
Ah, yes, the pox viruses. The true giants of the viral world. Highly complex brick shape, much larger, maybe 300 by 240 by 100 nanometers. Also enveloped. This physical diversity reflects their varied evolutionary paths, their entry strategies, and ultimately how they interact with and subvert our cells.
Okay, we've covered what they are, how they operate cellularly, the specific types causing cancer. Now let's pivot to the science. How do we actually diagnose these viral cancers, and what are the treatments and challenges? What's the main hurdle in detecting these stealthy invaders, especially with that long latency?
The challenge of detection is indeed significant. A real diagnostic puzzle. Diagnosing viral infections, particularly oncogenic ones with those long latency periods and often subtle or no early symptoms, can be quite tricky. They rarely announce themselves clearly, making early detection a race against time and a test of our diagnostic tech. It's like searching for a tiny needle in a huge shifting haystack.
So what tools do labs use? Traditional methods might not always work for something so elusive, right?
You're right. It needs a sophisticated toolkit. Diagnosis involves several key methods, from traditional to cutting edge. Traditionally, there's direct microscopic examination. Pathologists look for characteristic cytopathological changes or distinct inclusion bodies inside infected cells.
Like visual clues.
Exactly. Abnormalities in the nucleus or cytoplasm that act like a signature of viral infection. Papillomaviruses and adenoviruses, for instance, can cause intranuclear inclusion bodies visible under a microscope, an initial clue. Another method, often the gold standard in virology, is virus isolation and identification, actually growing the virus in cell cultures.
But you said that's hard for some.
Yes, for many oncogenic viruses like HPV or KSHV, isolating them in culture is exceedingly difficult or impractical. They just don't grow well in the lab. This limits its use and pushes us towards more indirect but powerful methods.
Okay, so if growing them is tough, what about indirect methods, looking for the body's response?
Absolutely, we rely heavily on those. We use serological tests detecting virus-specific antibodies or antigens. These tests look for the body's immune response, the antibodies it makes, so for actual pieces of the virus itself, the antigens. Useful for EBV and KSHV, giving us a historical or active footprint of the infection. But increasingly, and maybe most importantly, the power of molecular amplification methods has become vital for sensitive, specific diagnosis.
Molecular amplification, like PCR. We hear about that all the time. And what makes it so powerful here?
Exactly. Polymerase chain reaction, PCR, is a cornerstone. It's incredibly rapid and highly sensitive for detecting and even quantifying viral DNA or RNA. Its power is amplifying even tiny fragments of viral genetic material, making them detectable. Used for herpes viruses, HPV, EBV, HIV, lots of them. PCR can also quantify the viral load, how much virus is present. Crucial for prognosis. Not just presence, but scale.
Beyond standard PCR.
We use DNA or RNA probes, specialized genetic sequences designed to specifically identify and quantify unique viral genomes, like for CMV, papillomavirus, EBV, molecular search and find missions. For a more advanced look, there's in situ hybridization, detecting viral genomes integrated or not directly within tissue biopsies. Crucial for persistent infections or cancer, showing exactly where the virus is in the tissue. A direct visual link. We can even use advanced genetic fingerprinting, analyzing restriction fragment lengths or electrophoretic patterns to get a unique ID for a viral strain, tracking its spread or evolution. Unprecedented precision.
So increasingly sophisticated ways to find them, identify their fingerprints, but treating them. That seems like a whole other challenge since they hijack our own cells. What are the fundamental hurdles?
The fundamental challenge is their very nature. Obligate intracellular parasites. They need our cellular machinery to replicate. This makes it incredibly hard to design antivirals that specifically inhibit viral functions without harming our host cells.
Stopping the intruder without demolishing the house.
Exactly. Trying to disable the intruder using your own furniture as a shield. Plus, viruses mutate rapidly, developing resistance to drugs, an ongoing challenge. Long-term antiviral use can also have side effects. It's a careful balance.
And specific treatments. Are there effective antivirals for all of these, or is it still developing?
Very much a developing field with varying success. For HPV warts, they might regress spontaneously or be removed physically or chemically, surgery, freezing, burning, topical agents. Prevention is key. Avoiding contact, safe sex. And crucially, the HPV vaccine has been a monumental public health success, dramatically reducing cervical and other HPV cancers by preventing infection. A powerful testament to preventive medicine.
The vaccine is huge. What about others? HHV8.
For HHV8, Kaposi's sarcoma, especially with HIV, antivirals like foscarnet, ganciclovir, cidofovir show activity against KSHV replication, helping manage it. Suppressing viral load helps control the cancer.
Hepatitis B.
For chronic HBV, carriers might get long-term interferon therapy or direct-acting antivirals, like nucleoside analogs. They inhibit replication, reduce viral load and inflammation, lowering liver cancer risk.
And hepatitis C. You mentioned breakthroughs there.
Yes, the landscape for HCV is revolutionized. Older interferon ribavirin therapy is replaced by newer direct-acting antivirals, protease inhibitors, etc. They achieve remarkably high cure rates, often over 95%.
Wow, 95% cure rate.
Which directly cuts the risk of liver cancer for those treated. Vaccines are also being explored. Huge progress.
That's incredible. What about HTLV? Are there specific treatments?
Interestingly, our sources don't mention specific, widely available antivirals targeting HTLV-induced leukemia. Management usually focuses on treating the resulting cancer, the ATLL, often with chemotherapy. It highlights that some viral infections still pose big challenges for targeted antiviral therapies to eliminate the virus or prevent cancer, an active research area.
Yeah, and HIV. Management, not cure, right?
Right. HIV causes opportunistic cancers via immune suppression. The infection itself is managed with highly active antiretroviral therapy, HAART. A powerful drug combo, nucleoside inhibitors, protease inhibitors suppressing replication to undetectable levels. HAART drastically prolonged life, reduced complications, including some HIV-associated cancers, but the virus persists intracellularly in latent reservoirs. It's not eradicated. And crucially, despite decades of research, there's no widely available preventative HIV vaccine. Prevention via behavior and PROATEP is critical.
A stark contrast with the HPV vaccine success. Shows the hurdles.
Immense hurdles for certain complex viruses.
So treatments manage viral load or fight resulting cancers, but it's a constant battle. Prevention is key where possible. Which leads us to our final section, surprisingly relevant to everyday life. Implications even at the dentist's office.
Yeah, it's fascinating how deep microbiology runs through all healthcare, dentistry included. Understanding oncogenic viruses is crucial for dental professionals, not just academically, but for direct patient care. And by extension, it's relevant for all of us. Our oral health can be a window into our overall systemic health.
So how do viruses causing, say, cervical or liver cancer actually show up in your mouth? Why is dentists positioned to spot them?
There are direct significant links. HPVs, for instance, are explicitly connected to oral cancers, especially squamous cell carcinoma. Also cause oral papillomas, focal epithelial hyperplasia-specific mouth lesions. A dentist can be the first to spot these potential issues.
Okay. Well,
Epstein-Barr virus, EBV. Beyond nasopharyngeal carcinoma and Burkitt's lymphoma, it can manifest distinctly in the mouth as oral hairy leukoplakia.
Hairy leukoplakia.
Yeah, a fuzzy white patch, usually on the sides of the tongue, almost exclusively seen in immunocompromised people, especially with HIV. Its presence often indicates a poor prognosis for AIDS progression, an early warning sign a dentist might spot.
Wow, so seeing that could point to a serious systemic issue.
Exactly. It signals a much broader immune problem. Then there's HHV8, causing Kaposi's sarcoma. It's a well-recognized oral manifestation in HIV patients, purplish, raised lesions, and hepatitis C, HCV. Primarily affects the liver, but surprisingly can present orally as lichen planus, an inflammatory condition, and might increase oral malignancy risk in some. The mouth truly is a diagnostic canvas.
So dentists really are on the front lines sometimes, acting as first responders.
Absolutely. A vital role. Oral signs can be the earliest indicators of systemic infections like HIV or other immune issues. Dental pros need detailed clinical oral microbiology knowledge to diagnose these. It becomes integral to the patient's overall treatment plan, often needing collaboration with doctors. Diagnosis can involve microscopy, looking for things like multinuclear giant cells, or increasingly, those modern sensitive PCR methods are used in advanced dental diagnostic labs.
This also makes me think about infection control at the dentist. Things changed dramatically after HIV emerged, didn't they?
Spot on. The emergence of infections like HIV in the 80s and 90s literally revolutionized and significantly increased the regimentation of infection control in dentistry. Before HIV, protocols were less stringent. Understanding bloodborne pathogens completely changed the game. Affects patient safety, the rigorous protocols in every office today. Dentists must be incredibly well-versed and consistently adhere to these measures to protect themselves, their staff, and crucially, their patients. These visible precautions also address patient concerns, which were very real back then.
Makes sense. Bloodborne viruses are a big concern.
Particularly concerning due to work involving blood and saliva. Hepatoviruses, like HBV, hepatitis B, pose a significant risk via Sharps injuries. That's why HBV vaccination is strongly recommended, often mandated, for dental staff. Hepatitis C, HCV transmission in dentistry isn't unequivocally proven, but HCV RNA can be present in saliva. Warrants extreme caution and rigorous universal precautions treating all fluids as potentially infectious.
And I remember hearing something really specific about prion diseases in dentistry.
Yeah.
Even harder to deal with than viruses.
That raises a critical point, a truly fascinating, almost chilling detail. Prion diseases like variant Creutzfeldt-Jakob disease, VCJD, they're infectious proteins, totally different from viruses, and they are exceptionally, terrifyingly resistant to routine sterilization like autoclaving.
So standard sterilization doesn't work.
Not reliably. This unique resistance needs special, more aggressive decontamination, or often using disposable instruments to prevent any transmission risk, particularly for endodontic instruments, root canals, because the dental pulp has neurological supply and prions affect the nervous system. A totally different challenge, but powerfully reinforces the need for extreme vigilance and adaptable protocols.
So all these pathogens, oncogenic viruses, common viruses, even prions, mean a constant need for strict protocols and evolving science.
Precisely. The microbiology landscape in healthcare is dynamic. Other viruses like HHV6, common in saliva, rubella, spread by nasal secretions, further reinforce the need for standard infection control. So the fundamental principle in dental practice, standard infection control precautions, is critical. Treat all patients as potential carriers, all body fluids except sweat, as potentially infectious. It underscores those unseen, rigorous layers of safety meticulously built into modern healthcare, protecting you, the patient, and the providers.
So reflecting on this deep dive, what does it all mean? We've gone from the microscopic world of viruses and genes, their intricate subversion, to their profound impact on health, even routine dentist visits. An incredible journey into a hidden battle.
It truly has been. The aha moments are plentiful. Realizing that significant percentage, 10 to 20% of human cancers are linked to common, often unseen viral infections, or understanding the intricate, almost intelligent ways these viruses evade our sophisticated immune systems, turning our own machinery against us, reveals the hidden complexities, the delicate balance of health and disease.
And for you, our listener, being well informed about oncogenic viruses isn't just academic, it empowers us to understand public health initiatives like vaccinations, make informed decisions about medical advice, appreciate hygiene practices, safety protocols, all with greater clarity. It demystifies complex threats.
Yeah, and connecting this to the bigger picture, it really highlights that ongoing, relentless evolutionary race between viral adaptation and human medicine. We've seen how sophisticated these viruses are, adapting to persist and subvert. Considering their long latency, silent for decades, and their astonishing evasion tactics, it raises an important provocative question for us to ponder. What does it truly mean for us to be virus-free in a world where these stealthy disease architects can linger so long? And how might future breakthroughs, advanced diagnostics, maybe genetic editing, allow us to detect or even counteract these silent threats years, decades before cancer even appears?
A powerful thought for the future of medicine.
A future where we might predict and prevent cancer in ways we're only just starting to imagine.